Hello everyone. This video is for November 11th or 12th, and we are going to do the following things today. We're going to start with the DNA replication quizzes. A few weeks ago, we did one for protein synthesis, very similar to Kahoot. It's like a review game, and that's how we'll use it. Though we will do it in class. If you're watching this because you were not able to make it, there is a link for you to do it on your own so you can get those participation points. After that, we will move on to mutations and we will take some notes. After that, we will practice what we learned in the notes on a worksheet, and we'll kind of put together protein synthesis and mutations kind of all in one, so you get a little practice with both. To get to the review quizzes, you'll go to Canvas and go to Modules. Like usual, you'll go all the way down. You'll go all the way down to where it says DNA Replication Review, in case you missed it on Wednesday or Thursday. And you'll go to this link, enter this code. I'll do it really quickly so you can see what it looks like. Put in that code, join. Make sure to put your name in there and start. It'll be like a little game. It's a review game. Once it's done, just like Ed Puzzle, you don't have to submit anything. It'll just let me know when you did it. So next up is the mutations notes, and we will take notes and you will turn them in for some hopefully easy points. So once we are on Canvas under modules, it'll be right underneath what you just did, that replication review. You'll click on it. So we will take notes together in class. I'll upload the notes and another video of them to help you. I might just add it to this one. So what you'll do is go to this document and make a copy. This is now your copy of it, so you can actually write in it. And as we go through the PowerPoint slides, you will write in all the answers to these questions. And when you're done, you will turn them into Canvas. So just like usual, you go to the Submit Assignment button, go to your Google Drive, find this document, and submit it. All right, so now we are going to move on to our mutations notes. So as you're following along, you fill out those questions on the document I gave you. So the first slide will answer the first question. What are mutations? So mutations are changes in the structure of a gene. We talked a little bit about genes, but I've also included the definition. A gene is a piece of DNA that codes for a specific characteristic. So here's just a section of the DNA strand. Not every part of the DNA strand codes for the same characteristics. You have different parts that code for different things. So a mutation is anytime you see a change in the structure of that part of the DNA strand. So that's going to answer your first question. A little extra information is just that mutations can occur in somatic cells. Somatic cells are just your body cells, like muscles, skin, brain cells, those things. So mutations can happen in those or in gametes, your sex cells like eggs and sperm. So you can get a mutation sometime in your life, something like cancer, or you can pass on a mutation from a parent to offspring, maybe something like diabetes or something of that nature. So number 2, are mutations harmful or helpful? There actually can be both and neither. Some mutations can actually improve an organism's survival and be beneficial. For example, sickle cell anemia is not a mutation that's beneficial, and that's when your red blood cells go from circular to more sickle or crescent shaped. Can really hurt your blood flow and cause a lot of issues. But for those who carry the gene, they can actually be less susceptible to a disease called malaria, which is an interesting mutation for sure. Some mutations are definitely just harmful. In the case of cystic fibrosis, where there's a lot of mucus buildup in your airways, that's definitely a harmful mutation. But interesting enough, most mutations don't have any effect on the organism at all. So there are two types of mutations that we'll talk about. Today we'll focus on gene mutations, but next week we will talk about chromosomal mutations. So a gene mutation is a change in the nucleotide sequence of a gene. Remember that small segment of the DNA that codes for a specific characteristic? Most of the time, they only involve a single nucleotide. And these mutations can be due to a variety of reasons: copying errors during DNA replication or during transcription and protein synthesis. It could also be due to things like chemicals and viruses and things of that nature. So that answers number 3. So there are different types of gene mutations. We'll talk about point mutations, substitutions, insertions, deletions, and frame shift mutations. So for number 4, what is a point mutation? A point mutation is when a single nucleotide is changed. And this could look like a deletion, an insertion, or a substitution, but it's just one nucleotide. So in this picture down here, you see an original sequence. And then down here at the bottom, you see that instead of that T, there's a C. So that would be your mutation. It changed. One changed. So we'll look at how these different point mutations kind of affect your DNA sequence. So here we're going to look at a sentence, so it makes a little bit more sense to our brain. These words are 3 letters long, just like codons are 3 letters long. So we're going to see how that affects, or how that is affected by mutations. So, all right, we'll take this out so we can just do substitution. Okay, but a substitution, just like what it sounds like, is when one letter is removed and replaced with a different letter. For instance, if we want to take this C and switch it out with another letter, our sentence may look a little like, the fat rat ate the rat. So here is your substitution. Turn from cat to rat. It seems very small. It doesn't really affect the sentence as a whole. Changes the meaning a little bit, but it doesn't completely derail the sentence. So this is a substitution. Okay, this will be for number 5. Next, we'll talk about an insertion. An insertion is exactly what it sounds like. It inserts a base in there. But keep in mind that we keep having to read in this 3-letter format. So let's say we add a letter around the same spot that we substituted. So right here between the C and the A in cat. So we have the fat we're going to add an A. But again, we're only reading in 3, so then it becomes I'm going to make this a little longer. Okay, so we added an extra A here. And you can kind of see how it starts off normal, the fat cat, but the rest of the sentence starts to not make any sense. So this pushes this reading frame over a little bit. So instead of the fat cat ate the rat, this mutation will read the fat ka tat f era t, which does not make sense. that's an insertion, just inserting a single nucleotide into this sequence. This will be for number 6. The last one we'll talk about is deletion. So very similar to insertion, but the opposite. You are deleting one nucleotide. So let's stick with the same one we've been playing around with. And let's say we're going to do the C. So we're deleting it. And again, still reading in 3's, so our sentence will look a little like this. Okay, this is where the C would have normally gone, but we took it out. So now the sentence reads, the fat atta tet her at. Again, just like an insertion, before that mutation, our sentence looks good, it makes sense. After that, though, it kind of goes crazy. This will be for number 7. So both an insertion and a deletion are known as frame shift mutations. So remember that inserting or deleting one or more nucleotide can change the whole protein. It changes the reading frame, like changing a sentence. So you saw that in an insertion, the reading frame kind of shifts to the right, and in deletion, it shifts to the left. Just like our sentences stop making sense, those amino acid sequences are going to change and make the proteins be built incorrectly. They won't be the right protein, and they won't be built right. They'll be mutated. And this will be for number 8. So that kind of looks like here is you have our DNA sequence. So we have methionine, glutamine, alanine, and glycine. And in this mutation, we took this G out. So then our reading frame shifted. So then we started with AAG, CAC, GT, just like down here. And then our amino acid sequence becomes methionine, lysine, right? That's already wrong. So this protein will not be what we need because of that mutation. Here's an example of an incorrectly made protein in the event of cystic fibrosis. So here we have this protein that's made normally. It allows for those molecules to move freely in and out, and it helps keep our airway normal without that buildup of mucus. But because this mutation caused for this protein to be built incorrectly, it can cause a lot of issues like mucus buildup on these membranes and make it a lot more difficult to breathe. Now, after learning all of that about point and frame shift mutations, which one do you think would be potentially more dangerous? And tell me that for number 9. Last on our agenda today is our DNA mutations practice worksheet, which you can find on Canvas. Just like everything else, it's going to be organized in modules. All the way down. It will be DNA mutations practice. Okay, so you'll need to make a copy of this document. While it's loading, I will note that you will have to use this codon chart. I did not include a wheel, but that is on my list of things to do. If you do prefer the wheel, a quick Google search for codon wheel can help out significantly. So I'm going to let you know what happens on this page. So your directions are to transcribe and translate the original DNA sequence, which is this one. And then you're going to do the same for each mutated DNA sequence. And then determine the consequence, if there's any, for each mutation. And then you'll highlight your answer or your choice for each question. I will do the DNA sequence here to help you kind of figure out how to do it, and the first mutated sequence. I do also want to point out that every time you do a new mutated DNA sequence, you'll compare your mutated sequence to the original. That's how you know what kind of mutation happened and what the consequence is. So first we have to transcribe our DNA sequence into mRNA. So here we'll start with TAC. What bonds with TAC? It is AUG. ACC, what bonds with that? UGG. Keep going. AAC. CGC. UGC. And UGA. So here's our mRNA sequence. And it's already kind of put into codons, which is nice. So I'm going to split screen this just for ease of looking at both screens. Then we'll use our codon chart to figure out what amino acids these code for. So AUG, AUG is methionine. UGG, UGG is tryptophan. AAC is asparagine. CGC, CGC is arginine. UGC, UGC is cysteine. And UGA, UGA is our stop codon. So this is our original DNA sequence, our original mRNA sequence, and our original amino acid sequence. So pretend this codes for a very specific protein. These next sequences are going to be a mutated version of this original DNA sequence. So we'll do the same thing. We will transcribe it into mRNA. So what bonds with TAC? AUG. What bonds with ATC? UAG. What bonds with TTC? AAC. CGC. UGC. And UGA. So far, are there any very obvious differences between the original sequence and the mutated? Yes. So here also says highlight any changes. So I'm going to highlight this change right here, because we know in the original sequence this was a C, but it was mutated to a T, which gave us UAG instead of UGG. Then we're going to go ahead and translate those into amino acids. So AUG, AUG still being methionine. UAG, UAG is a stop codon. ACC will stay the same as asparagine. CGC will stay the same as arginine. UGC will stay the same as cysteine. And UGA will stay the same at the stop codon. So at the bottom, you'll see there's two questions. The first one is, what kind of mutation did we see here? It says, is it a point mutation where it's just a substitution, where we took one out and put in another one? Or did we insert or delete one whole nucleotide? Well, everything else after this looks the same. So it's safe to say that this is a substitution. That C was substituted out for the T. So I would highlight substitution as our answer there. For the last question asks, how did the mutation affect the amino acid sequence? So what's the consequence from this substitution? So your options are these. There's no change. Only one amino acid is changed. There's a premature stop signal. There's no stop signal. One amino acid was added or deleted, or all of the amino acids changed after the point of mutation. Now, the very obvious is instead of tryptophan, you had that stop codon. So I would say that this is a premature stop signal, and I would highlight that. Then you're done with this mutated sequence. Then you'll do that again down here for sequence number 2, transcribe, translate, and you'll always compare this mutated sequence to the original. What changed? And there are 5 total. When you are done, you will just go back to Canvas and submit assignment. If you have any questions, please don't hesitate to let me know.